Hello, and welcome to my presentation entitled How Gemma Collins Could Get Away with Murder. This is a molecular biology perspective on what I believe to be the first ever case study of what I like to call biological nepotism. Now, just as a disclaimer, this is entirely hypothetical. Let's get into it. So, Gemma has done something naughty. Someone is. Sometimes when criminals commit a crime, they are a little bit clumsy. They leave behind some clues, some evidence. This could be in the form of a strand of hair or a drop of blood. What this allows us to do is forensic investigators can collect this evidence and they can analyse the DNA inside to establish who has committed the crime. Which leads us to our first question: what is DNA? In its most basic form, DNA is a molecule that is the essential building block for all living organisms. Every single living organism has its genetic code written in the form of DNA. DNA is a double-stranded molecule that is in a double helix shape. It has a sugar phosphate backbone and it's made up of 4 chemical bases. These bases have names: adenine, A, cytosine, C, guanine, G, and thymine, T. A's and T's always bind together, C's and G's always bind together, and the combination of A's, T's, C's and G's will give us the code for our genetics. Every single human being has ever so slightly different DNA from one another, which allows us to identify individuals by analysing the DNA. We then move on to our next question. What is Gemma Collins? Well, for those of you unfamiliar with British pop culture, Gemma Collins, otherwise known as the GC, is a TV personality known for her starring roles in Towie, The Only Way is Essex, I'm a Celebrity, Get Me Out of Here, Big Brother. Such viral clips as, I'm claustrophobic, Darren. What all of this TV presence has culminated in is, I believe, Gemma's capacity to accumulate rather a lot of wealth. She's rich. Let's just bear that in mind. Back to the biology now, folks. So when we are analysing DNA, one of the first and most important principles in molecular biology is something that we call PCR, polymerase chain reaction. Here's a handy little diagram that I found on the internet. So we need lots of DNA to be able to analyse it, but at a crime scene, we might not have as much as we need. PCR allows us to generate the amount of DNA that we do need. So we're starting off down here with our little bit of DNA. Now remember, this is double-stranded. It looks a bit like a zip. The first step that we need to go through is called denaturation. This is where we unzip the DNA, which leaves us with 2 single strands. We do this step by heating the DNA up to high temperatures. This allows us to unzip it. This DNA is floating in a solution with other bits and bobs. Some of those bits and bobs are called primers. These bind to the DNA. And then other bits and bobs that are in our little solution are A's, T's, C's and G's. Throughout the rest of the PCR process, we bind our primers to the DNA and then we bind our A's, T's, C's and G's to their corresponding partners in each of these strands. This then leaves us at the end of our first cycle of DNA with 2 DNA molecules that are identical to our initial 1 starting point. We can then repeat this process again and again and again and again, going from 1 to 2 to 4 to 8 to 16 to 32 to 64 to 128 until we have enough DNA to analyse. But what do we know about science? Well, we know that it doesn't always go right. Things just go wrong. And I want to talk specifically about one of the problems that we face in PCR. So we know, I've already told you, that A's and T's always bind together and C's and G's always bind together. But actually one really key principle that I want to highlight, we see our bonds here. This is how they're bonded together with hydrogen bonds. And we see in our A and T they're bonded together with 2 hydrogen bonds, whereas our C and G, we have 3 hydrogen bonds. This is important. When we have more bonds between things, it takes more energy to break apart. So it's going to take more energy, more heat in our first part of the PCR reaction to break apart our C's and G's. Now this is pretty negligible, but what happens when we have loads and loads of G's and C's together in one region of our DNA? Well, this is called a GC-rich region. GC-rich regions are much more difficult to do PCR on because you need more energy, more heat to break them apart, and also GC-rich regions of DNA can form all these weird and wacky shapes, structures, which make the PCR process as a whole more difficult. And here is just an example of a paper talking about this. This is published in Nature, which is the vogue of scientific publications. So what does this mean if our PCR won't work on our GC-rich regions? Well, what it means is that we're not going to have that DNA evidence that we need to incriminate Gemma at the scene of the crime, which is where we return to now. She's done it. She's done it. And off she trots. Oh, hang on. Are you seeing what I'm seeing? Gemma's been careless. She's left behind some of those luscious golden locks, which means that I can take them back to the lab to analyse them, which I shall do. However, I just can't get my PCR to work. It's just not happening. It's failing at every step. Let's revisit some of the things that we know about Gemma Collins. You see, folks, there's a reason that I call this the first ever case study of biological nepotism, because Gemma was always going to get away with this. The PCR was never going to work. It was always going to fail. And you want to know the reason why Gemma Collins could get away with murder? Well, it's because GC rich. Today marks the 1-year anniversary of me making TikTok videos. I know that this one is slightly different to my usual, more educational dementia content, but I wanted to do a little fun thank you for all of you for showing me so much support over the last year. Normal service shall resume shortly, but for now, thank you so much. I hope you've learned something about DNA or PCR or Gemma Collins that perhaps you didn't know previously. Have a lovely evening. Bye-bye.